Les, you guys don't realize how lucky you are to have the tech transfer team, and Les, that type of research is what is the most important research to me, that really practical applied research in the field, incredible research. Well-designed trials. My hat is off to you guys. That's great stuff that you're doing, and I pay very close attention to the details of what you're doing. So let me start off. The day before I flew here, I was out in the almonds. That's what the California almonds look like on that day. We had to drag all those hives. The entrances had been underwater. I drug them all out of the water, and we got lots of rain in California. So, come on. Come on, remote. Let's try this on. Okay, there we go. When you have that kind of humidity in the almonds, then they spray fungicides. We took this picture also in a test orchard. And this is the University of California experimental almond orchard, which by just a fluke 35 years ago, I got the contract to pollinate. So I pollinated all the experimental almonds in California for 35 years. So I worked with the orchard manager. So the question I had is, we're getting a lot of complaints by beekeepers about the sprays of the fungicides, and especially the tank mixes of a fungicide with an insect growth regulator. And I'll tell you right now, you do not ever want to have a tank mix of an insect growth regulator and a fungicide. And there's two of them that are especially synergistic. Our hives are just sprayed right over with this. We don't normally see a whole lot of problem. The growers all know the best management practice recommendation is to spray at night, but nobody wants to work at night. So when we go to robotic spraying in the future, that'll be better because they can do it at night. So what I've been trying for the last 3 years to quantify the effect of the spraying on the foraging activity of the bees. So we went out, there I am, in the lovely weather, setting up monitors. So these are monitors on the hives right here, entrance flight monitors. A couple of different companies have tried to work with me. Hugh Evans from Arnia, who does the hive monitors, really pulled through, has worked very closely with me, and custom made these. And he came out, flew over from Italy, and spent the time with me to install these monitors in the hives. So they look like this. We're catching rainfall, temperature, humidity, weight underneath the hives, and then this are interest in and out counters. Counts how many bees are exiting per minute, how many bees are returning per minute. And this is the type of graph that we're getting here. So the black line is the bees that are exiting, and the orange line is the bees that are returning. Now you lose bees every day. So a rapidly growing colony is going to lose 500 to 1,000 bees a day just from aging. So the orange line is always going to be lower. What we want to see is whether or not we can detect effect from fungicide spraying. So we have 2 hives set up with monitors in each of 4 different orchards, and all the orchard managers are all coordinating with me with their spray reports, the hour that they're spraying and the day that they're spraying. So we'll see what we can get from there. So this is research that's going on today, right now, in the almond orchards. Okay, so last season, I did a few trials, and I had to decide we don't have enough time in an hour to cover all of these. So I'm going to blast really fast through a few of these, and I'll save the oxalic acid for last because I've already published that research, and you're able to see that research results on my website. The other stuff has not been published yet, so I will show you what we're doing with that. Big question with beekeepers, especially up in California where we have these dry conditions, and if you don't feed pollen sub, it's very hard to get your bees to go into winter into good shape. We do a lot of supplemental feeding. Many of us feed in the fall around the 10 to 15 pounds of pollen sub every fall to every colony to build them up to go into winter in good strength because we don't have the fall pollen flows. I did a trial in 2013 comparing different pollen subs that are on the market with a positive control of patties, 1 pound patties made of natural bee collected pollen mixed with sugar syrup, and a negative control which was just sugar alone with no protein. So that's to confirm that the experimental design is valid. You expect the natural pollen to perform the best and the negative control to perform the worst. Here's the frame strength of bees over time, going from August until almond pollination in February the next season. So what we found is without the supplement feeding, they were not strong enough to go to almond pollination. And with feeding pounds of natural pollen, they did quite well. The interesting thing was 2 of the pollen subs actually initially outperformed the natural pollen, which was a surprise to us. But then the natural pollen seemed to turn around and bypass it. That indicated there's a limiting nutrient in these, and the higher protein content of these probably initially gave much more colony growth, but after a while they ran out of something, and protein alone wasn't enough, and then the whatever was in here outperformed them there. So we get to the concept of limiting nutrients, which means if you're going to build a structure out of materials, repeating structure, you can build until you run out of the first material. Even if that first material is a simple nut or a simple bolt, you can't go any further. It doesn't make any difference how much other structural materials you have, because you need everything together. So like if you're trying to raise kids and you give them every nutrient there is, except for vitamin C, they're not going to grow. They're going to get scurvy, they're going to die. Where all it takes is that one limiting nutrient to allow the growth. So beekeepers that focused on amino acids and protein and all that, I think we're wasting our time with that, because there's some other limiting nutrient there that is preventing us to use all that protein. So any protein that's in the patty beyond the limitation of limiting nutrient is just totally wasted. It's just excreted by the bees. So we need to figure out what that limiting nutrient is there. So we don't know what it is. So a few things are happening. One is that as we humans use more fossil fuels, dump carbon dioxide in the atmosphere, it lets the plants grow faster. But that growth is only for carbohydrates, for starches, for sugars, for cellulose. The plants don't make any more protein, and they don't bring any more minerals up into the plant. So our food becomes more calorie rich, but less nutritious overall on a weight basis. And the livestock industry is very much aware of this. And 1/3 of the human population is dependent upon wheat and rice as their major food source. And wheat and rice are becoming less nutritious every year. More calories, but less other limiting nutrients. The other one is the zinc zinc. And not only does the carbon dioxide reduce the protein, but it reduces the zinc in the diet. And this recent paper found out how much zinc bees actually need. And what it is they need roughly, the nurse bees need more zinc than the field bees do. But typically, the bees are going to need 60 to 75 parts per million of zinc in the diet. And surprisingly, not surprisingly at all, that's exactly what you put in any other livestock diet. So that seems to be a common factor, that anything growing needs about 60 to 75 parts per million of zinc. So pollen is the perfect food for bees. So let's take a look. This is pollen collected from Poland. About half of what they need. Pollen from Korea, around the same area. Pollen from China, low. So the limiting nutrient for bees may not be the amount of protein in pollen, it may be the amount of zinc in pollen. And then this recent study of 2016 showed that when they collected goldenrod pollen from museum specimens, that as carbon dioxide has gone up in the atmosphere, the protein's gone down. Since 1960, there's about 1/3 less protein in goldenrod pollen, which means that bees have to work half again as hard just to stay even with where they were back in 1960. You have to have half again as many pollen loads coming into the hive, and the nurse bees have to eat half again as much pollen just to get the same amount of protein that they got back in 1960. So this is invisible, but it may be a reason that keeping bees healthy and alive is a little bit harder today than it used to be, besides Varroa and all the other stuff that's happening. The other one is work by Elton Herbert and Svoboda back in Shimanuki back at the USDA lab. Elton Herbert did this incredible research on bee nutrition, artificial diet, and he died unexpectedly at an early age, and all this research was just dropped. So it's fascinating research, and what they found is the greatest survival of bees is, for cholesterol. To you guys, your cholesterol in your diet doesn't mean anything. Your body makes its own cholesterol. So that's why this whole thing about don't eat eggs for your cholesterol is nonsense. Your body makes its own cholesterol. Insects don't. Insects, sterols are vitamins to them. They can't make their own. They have to get it from the diet. Each different species of insect may need a different kind of sterol for their sterol needs. For honeybees, it's 24-methylene cholesterol is the main sterol that they need. The other sterols, plant sterols, don't do much for them. They need to have that 24-methylene cholesterol. When we look at 24-methylene cholesterol, in the pollen, in the adult bees, and in the pre-pupae, you notice it comes in, let's look here, it comes in at 5.1 parts per million in the pollen, and when they measure the pre-pupae, it's up to 43. Down here, it comes in at 15, up to 59, 1.5, up to 52. What Elton Herbert Jr. found when they used radioactive tagged 24-methylene cholesterol is the nurse bees concentrate it in their bodies. And then when there's not enough coming in the pollen, they take it out of their own bodies, put it in the jelly, and they give it to the pupae. And even if there's none in the diet, they can do that for a while, keep that going. So I suspect that may be what was happening in the pollen patties, that when they got the high-protein artificial patties, they were able to pull the 24-methylene cholesterol out of their bodies. But after, what Herbert found is after 2 generations of brood, then the nurses ran out, and then the colonies would go down. So that fit the data, the results that we saw in 2013. So I've searched for years for a source for 24-methylene cholesterol that was inexpensive. And finally, when you look at all these are all vegetable oils, corn, cottonseed, borage, evening primrose, canola, palm, peanut, sesame, safflower. There's only, these are all different plant sterols that are found in these. For 24-methylene cholesterol, borage oil is the only one that has a high amount. Now, 5 years ago, that was hard to get a hold of. Nowadays, on Amazon, you can buy borage oil really cheap. I found a source for the borage oil. I also hooked up and helped get some funding to Priya Chakrabarty and Ramesh Sagili over in Oregon. They had some artificially concocted some 24-methylene cholesterol that they, for like $10,000 for a couple of grams. They can now use that to calibrate the instrumentation. So she's doing all the analysis of 24-methylene cholesterol for me in this particular trial. So I tested 4 different diets. One standard patty that we have used for a long time. One standard patty supplemented with zinc and the 24-methylene cholesterol and borage oil. I applied the zinc in both organic and inorganic form in the recommended proportions. Then a positive control. Now this time, instead of feeding 1 pound patties of pollen, I fed equal protein content. So I got a natural pollen from 3 different sources, sent it in for analysis, and to find out how much protein there was in it. And it's a lower protein content than the standard patties. So these were now larger than 1 pound patties, but they were identical for the amount of protein. And then I also mixed these up with the identical amount of sugar as in these patties. Then we made a negative control that had no protein in it. It was just sugar, but the same amount of sugar that is in these 2 patties. Okay, you with me so far on we set it up? Okay. Then we set up 72 hives. These were nukes that were had grown to about 6 frames or so. When everything goes dry in August in our area, in a yard where historically the bees all just go downhill if we leave them there. So very little chance of growing them up to pollination strength. So then what we did, we emptied these hives, we broke them down. Now the way I like to start out these trials is I only leave 5 frames in the middle of the box, put 3 frames with brood in it, and then 2 frames with some honey on the outside. And then we can take all the lids off and see how much coverage we get on the outside of bees. Very quick assessment method for equalizing colonies for strength. So we want to get them all so they have the same coverage of bees on the 2 outside frames. And what we do is we'll spend a week going through them and just shaking bees from one hive to another through a queen excluder, and until we get all the hives equalized for strength to start with. Then we had to mix up our sham patties and our control patties. So here we are mixing up take the pollen out of the freezer, mix it up with the right amount of sugar syrup, and then chop them up and then weigh them so they all were right. And then take them out there. Randomized block design, 4 hives on a pallet, each one facing in a different direction. Randomly assigned which got which type of patty. We only ran 13 each of the positive and negative controls, and 25 each of the 2 patties that we were trying to compare of the for treatment for difference in the treatment. And at the end, the negative control patties did really well, really poorly. The colonies barely grew. This is after 26 weeks, 11 feedings, 19 gallons of sugar syrup. It was just this was total artificial feeding to grow these colonies up. and very little growth in the sugar controls. Very nice growth in the pollen controls. They all went to almonds at very nice strength. This was before almonds. They're already 8.5 frame average. They would have easily been over 10 frame average by the time we go into almonds. And the treatment, the standard and the supplemented diet, no benefit. So beautiful hypothesis just shot down entirely by actual data. So what this is telling me is, you know, I still feel that we should, oh, I also had the standard patty tested. It had about 1/3 the amount of zinc that it should have had and 0 24-methane cholesterol. So I'm really surprised by this result, but this is the hard data from the yard. So my guess is, one, we probably should supplement it, but two, we still have a limiting nutrient that we have not yet identified because we still have the natural pollen patties outperforming at a protein-to-protein basis the natural patties, which means we have much of the protein in these patties is just wasted. Protein being the most expensive component, we're not utilizing all that protein in those patties. Okay, so there's my conclusion right there. But you know, you could be, I had so convinced myself that it was gonna, you know, I found the answer, and every week it just got worse and worse because we just didn't see an effect. Okay. Another one, I'm doing the Beekeepers, guys, we've had Varroa for over 30 years, and we're still using fly swatters and Band-Aids to deal with this problem. You know, we need to hand the job over to the bees. We need to all be running bees that are mite resistant. And a number of groups across the world are finally realizing this. And they often make it really complicated on how to do it. And the problem with that is the queen producers aren't interested in doing something complicated. They need something really, really simple in order to if somebody sends them resistant stock queen mothers, they need to keep that from getting diluted out after a couple of years in their operation. Plus, most of them want to use their own stock they develop. So I'm saying, well, can you just develop mite-resistant bees from your own proprietary stock of bees? And so I'm trying to show a simple method to do it. So I'm testing this out. This is not a done thing. I'm walking the walk. I'm applying this method myself to see what, and I'm publishing a progress report. And I may succeed and I may fail. But one of the persons who got interest is Dr. John Kiefer over in France. He's the guy who coined the bond method, the live and let die. And this is a successful, proven method. I visited John. He has the Varroa Challenge in France. He invites anybody to come over anytime and go into his hives and see how many mites you can find. And every mite you find, he gives you a nice little shiny copper French penny. And this was in November. It's kind of tough. I had to go through 6 hives dissecting brood to earn my penny. But I got my penny. But I can tell you right now, his hives untreated hives have very few mites in them. So he has demonstrated that live and let die works. No commercial breeder is ever going to use this method because 95% of your hives die, you know, in the initial years. So it's very, very expensive to do. So what John has done is he has now changed to the modified bond method, where he's suggesting that you don't have to let the colony die. You just only have to work with the genetics of the queen. So you can treat the other colonies, but apply strong selective pressure. So he got the French Queen Breeders Association to invite me over to demonstrate my method over there this last summer. And all I'm doing is I'm doing this very quick method of mite washing right here that takes very little time. What we did in 2017, we started with a little over 1,000 colonies, and at each assessment, each one of these is an assessment mite washing, all the hives that the mites had gone up in, we took out of as potential breeders, we treated them, so we didn't lose any hives at all. And then, so we went from 100% starting, 28%, 10%, 5% by fall, and the next spring we bred off of 20 hives, 2% of the hives. And we found out that doing this actually saves us money overall. It's cost effective if you know how to do the mite washes quickly enough. The problem is, how do you monitor for mites quick? Yeah, I prefer the alcohol wash. It's the most accurate, but it still takes a while. I'll show you in a second how we get around this. So I've come up with what I call smoking hot mite washing, where you go out with a crew of people. We started off first with a larger electric mite washer that just plugged into the truck battery. we use color-coded cups with matching tags that go on top of the hive. So you can just go lightning fast through a yard. So the crew of 4 or 5 of us, we can do a yard of 30 hives in 20 minutes from the time we open the truck doors to the time we close the truck doors. It's just boom, we're in and out, and we have a mite count on every hive. Let me see if I can show a 3-minute video here. Oops, come on. Down. Okay, hang on, I gotta do something here. Here we go. And see if we have sound. Okay, no sound. Okay, so what I'm doing is, this is in November. This is a potential breeder, had a mite count of 1, mite count of 1, mite count of 0, and now we're going to get the November mite count. So this is in a yard, commercial yard of bees. The other colonies had mites, but this one, without any treatment, has kept its mite level low up to this point in the season. This November count is when we do a lot of elimination of these colonies because you get that spike late in the season. So what you simply do is you pull it. We use double brood chambers. I don't want to get a brood frame. What I want to get is a frame near the brood. There's plenty of nurse bees on the frames elsewhere in the hive. And then we use a behavioral separation. If you simply shake them into a tub and you say the magic words, old bees fly back out of the tub and fly back home. You have to say the words, and they immediately, fly back. And you can immediately also tell if a colony is queenless, because if it's queenless, all the bees fly back immediately, because they're all old bees. If they all stay in there, they're all young bees. So now you scoop it up. Get an accurate scoop, use your fingers very carefully, you level off the cup, get them off the side. You will never, ever get stung by doing that. I do it thousands of times. I've never been stung once in the finger by doing that. You pop it into the cup, and now this is my latest, and I'll be trying to find a manufacturer. You carry it in the front seat of the car, and you plug it into the cigarette lighter, and it charges each time. This is prototype number 11 or something. I got a lot of time to find out exactly how this works. That one, the lid was leaking, so you'll see what I do with that in a minute. And then my assistant said, "Hey, why don't I just film you doing this?" We hadn't thought of it ahead, because normally, so I had to go get the jar of alcohol here. So I walk away. While this runs 30 seconds, you have close to 100% of mites washed off. We typically do 60 seconds for scientific work, and at 60 seconds, you have absolutely 100% of the mites off of there. So now I take this off, I take the lid off. I'm going to throw this lid away because it was leaking, so I'll just pop a fresh lid on. You lift the bees out, catch the alcohol, look inside, and you watch the edge right here as you pour it off, so you don't take any mites forward. You leave about a 1/4 inch of alcohol in there, put it on a white surface. You look down, and you do your mite count. And there was 1 mite in here. So you take it now and put it down, 1 mite. So our mite counts over the season were 0, 1, 0, and 1 with no treatments at all. Productive colonies made honey. Now looking out, you see the yard out here. This was a commercial yard. And I also know, because we ran another trial, this yard picked up drift of mites from another yard, experimental yard, got a fair amount of drift coming into it. So it had plenty of mite exposure. And I surprised my helper to take the camera out of her hand. Okay. So this mite washer revolutionized what we are mite washing. Oh, come on, how do I get back to, there we go. PowerPoint, research from current slide, and go back a few slides. Okay. So 2018, almost the same curve here. And you think, well, not much progress. But here's the deal. We started with 1,500 hives this year, and at the end, we have 70 hives left out of the 1,500 that have not needed any mite treatment, kept the mite level below 3%. 5 colonies scored 0 here, and 1 very productive colony was 0 the entire season, all in commercial yards. Now, the thing is, I didn't expect to see much progress this year because the queens that we select for have to mate with the drone pool from the queen's genes from the last season. So, since we selected 2% of the queens, that means that 98% of the genes in the operation that would be in the drones the next year would be did not come from the resistant colonies. So the red indicates the percentage of genes from the non-resistant colonies in the drone pool, and the blue would be the 2% that in the drone pool. So the matings, the first year would be this: So we wouldn't expect to see a whole lot of progress the first year. If you work this out through the years, the second year, then you're going to start to see 50%. And each year, what you're going to do is you're going to shift 50% of the genes in the drone pool because your queens supply half the genes each year, or all the genes, and the queen's genes are half the genes that are in the drone pool. So next year is when I'm going to look for excitement after we made it halfway, and then next year after that. So I'm thinking maybe I'm looking for 4 years of this before I really expect to see serious progress. And again, maybe I won't. This is an experiment. I'm not a salesman. I find out what actually works. Now, if it does work, I think this will revolutionize breeding because it's so fast and so easy. And we're not breeding for any specific trait. What we simply do is we define the job description for the hives, which is keep your mite levels low or your genes don't go in the next generation. And we just apply very strong pressure, that 98% selective pressure each year. So cross your fingers. Okay, then the other big question is, in 2017, we got a monster spike in mites after September into November, just as Les was talking about, but huge spike in mites. Eliminated over half of our potential breeders then. Hasn't been a whole lot of quantification of how much drift is coming in. This is some old work from Griatti in Italy back in 1992, and he showed that you can get up to thousands of mites coming in late in the season. Even if all the hives around have been treated and you don't see any colonies collapsing, you can get these massive mite immigrations. And the best I can tell is that that's what happened last year. And the question is should we think of every hive just like being a big sneeze of mites and deforming virus going out to the surrounding landscape? So I wanted to test this, okay? Plus I have my Varroa model, and in my Varroa model you can input what your mite drift coming in late season is. Well, I need hard data for what that mite drift is. So that's what I wanted to collect. So I had a number of questions about this drifting of mites and the drifting of bees late in the season. I'll come back to all these questions with the answers typed in a minute, okay? So this is a very complex trial, so pay attention because you're going to get lost if you don't do it. Here's my house right here, my orchard and garden, honey house, our truck yard, equipment yard, our bee yard up here. We have this clearing. I have solar panels and down here. I started off thinking I would use colonies with yellow queens and colonies with black queens and look for drift. And I realized just before we got ready for colonies to collapse that didn't work. So it was fast and furious the last couple of weeks of changing the method. I'll talk about this with Amazon packages arriving every day with critical things to test out, and it was frenzy, but then we wound up being able to pull this off. So what I wanted was I started with a dozen. We got 9 colonies to collapse from Varroa down here in the bottom. And then we those are donor hives that were mite donor hives collapsing. And then we had receiver hives in the middle of them, 16 feet away, 500 feet away, and we have some further away that I'll show you right there. And then we also put in 2 mite-free donor hives that were controlled to have 0 mites to compare the amount of bee drift coming from the control hives to the amount of bee drift coming from the hives that were collapsing from Varroa. Are you with me so far? Okay, because it gets better. So, I haven't used a synthetic miticide in my operations in 2001. So we put in both Apivar and Apistan strips, and they worked incredibly well. They just zeroed the mites. We don't have resistant mites in my operation. So that's how we freed them up. And then I looked back to how to track these bees, and there's a paper from '71 from my friend Dr. Norm Gary from UC Davis, and hooked up back with him. He gave me tips on this of how using magnetic retrieval of ferrous labels, so iron-bearing labels. So then I set up all the donor hives this way with electronic scale, so I could see whether they were collapsing or whether they were robbing, because there's no honey flow at that time. So any weight gain has to come from robbing. Magnetic interest trap. Now Dormant had to use a bunch of old school horseshoe magnets. Nowadays you get these cheap neodymium magnets in strips. So I cut a dado strip in here, and I slid in 5 neodymium magnets across here. So I'll show you the tags in a minute. This will lift a bee off its feet from an inch away and snap that tag to the strip. So there's no bee will get through with the strip. And then I set them low enough that the bees can put their feet down and wiggle, and it pulls the strip off their back and leaves it stuck to the magnet. Then we have put sticky boards on all these hives, so I can pull them out and look at the mite drop. So what we did is we waited till those strips zeroed out the mites in the hive, so there was no more sticky board drop and no mites in the alcohol wash. And at that point, any mites that show up the sticky board had to come from the outside because there was no mite reproduction within the hives. We counted a bunch of mites. There's Brooke going through there, counting the mites. This is actually from other trial where we had higher mite levels. The highest we ever got in a bi-weekly or semi-weekly count was around 45 mites coming in those 3.5 days. Okay, so here's the home yard here, 12 receiver hives, and then I have other bee yards around this is a half-mile radius, this is a mile radius, so we put some receiver hives out at some of these other yards to see if we could recover any drift of bees at greater distances. Here's a view from my back porch, solar panels down there. Here's the collapsing hives and down here, and one receiver hive, and then some mite-free receiver hives here, and then more this way back up to the upper yard there. And then these are these customized chairs I welded together the last few days as we figured out how to do this marking of bees. And then the 2 mite-free control hives. Okay, every hive was color-coded. Every donor hive was color-coded so we can identify the bees coming from them. And I did confirm by brood dissection, alcohol wash, that there was no mite reproduction going on in those receiver hives at all. Okay, so here's Anna. What we found out is the gals who grap queens for us are really good at doing this marking, and women who crochet are good at doing it. Most people are not very good at it. So I shifted over from an hourly rate to a piece rate, and Anna was very happy because she's never made that much per hour before. She marks she's almost as fast as me marking them. I still hold the record for doing it. I can brag on myself here. So I set up a table here. They have, for anesthetization, they have, we tried different things with carbon dioxide. We got a big tank of carbon dioxide. We tried all kinds of ways of manipulating the bees without anesthetization. None of them worked well. So we went to carbon dioxide anesthetization. And we found out when you turn the valve on the carbon dioxide in a plastic cup, you can easily explode the lid off the cup. So then I ordered these screw top plastic containers, and then I put a safety big hole with a cotton wad in there. That allows the gas to come out, but it won't explode the cup. And I put on my safety glasses, and my friend Brian was there. I hid behind a hive, and I showed it, and it didn't explode. He goes, "Great, let me try it, Randy." And he gets his own cup, and sitting down, so he sticks it here between his legs and gets the carbon dioxide. I said, "Wait, Brian, you know, when we're testing for explosives, we normally hold it further from our testicles." He goes, "Oh yeah, good idea." So when you're doing these scientific projects, it's so exciting. Sometimes certain things just shut off in your brain. So somebody responsible to always watch you when you're doing things. Okay. So then instead of using the big carbon dioxide bottle, I found out they sell these little tiny tire fillers for bicycles. And you take a little CO2 canister, and the little valve fits over the valve stem, and you can fill up a tire. So we use those. And then I took the little needles used to fill soccer balls and saw the tip off of those and used that for injecting the carbon dioxide. We then punched out these little steel discs with a hand punch, one at a time, over 6,000 of them, and then glued them on double-sided tape on these cards, and then hand painted every one of them a different color to match the hives. And then I set up, just like marking queens, how we used to do it with tester's paint. We tested out a number of different kinds of glue, came up with a quick drying glue with a little dauber that we could use to glue these tags on the backs of bees. And we gave them just a very quick shot of carbon dioxide, and then take the lid off immediately, and the bees recover. But all the young bees just sit there looking up at you saying, "Okay, take me next, take me next." So it's very easy with light forceps to pick them up. We went through a whole bunch of carbon dioxide. Here's Brooke at her table showing, I took some pictures to show you how she's marking a bee here right now. We found out by trial and error, lots of trial, even more error. You take a thin piece of plastic and cut just the right shape V and then put masking tape around it. You can lower it over the bee's petiole, their waist, and it pushes their wings out of the way and exposes their abdomen, and the bee is not harmed. And you can take the dauber, put a drop of that fast-drying glue right there, and then you pick up with a pair of forceps one of the magnetic tags and just drop on there and tap it down. And then you grab the bee with soft insect forceps on the thorax, lift that plastic, and just place it back on the top bar right next to the little table. And you can just bing, bing, just very quickly mark these bees. And the bees sit there for a minute, then they walk down and they act totally normally. They go down to the brood chamber, and since they're all young bees that we're marking. Now, we marked them just as the colonies began to collapse. When I started to see the bees with deformed wings and I saw the brood starting to die, and so we marked a bunch of relatively young bees that time to see what those bees would do at the time of colony collapse. And then what was really neat is you wouldn't see any bees at all for the next week, and then suddenly on the landing board, all those bees just start moving out and change from nursing duties to guard duties, and then hang out for a few days on the entrance. Totally normal behavior. So there's like 7 of them here in this picture, all at the same time. So all these colonies did that. And then you see bees start coming back from foraging flights. And then we start seeing them up at our syrup tank. And when we spilled some pollen on the ground, a red bee showed up, and pretty soon a bunch of red bees showed up. So normal recruitment. And so we didn't see any change in behavior, any mortality of these bees. And we glued the tags on the abdomen, so we didn't change either their magnetoreception or their weight balance when they're flying. We put the weight back where they normally carry weight in their crop. And then at collapse, we went out and we would measure how many frames to cover the bees, so for a bee count approximation in every hive, and did alcohol wash for mite infestation rate, so you can see the mites on here. So at collapse, these bees had plenty of mites on their bodies, but very different rates. Some of them collapsed at a lower mite level than others, kind of surprised us. Usually we see it with alcohol wash, they start to collapse between 45 and 60 mites on an alcohol wash of a half cup of bees. But some of these collapsed at a lower rate. And then we collected a ton of data. And then, so I'm still working through the data. I am not done yet. So what I did is I color-coded some of these charts for you. So the red is the receiver hive that was right in the middle, less than 15 feet of the collapsing hives. The blues were 60 feet away. The greens were around 500 feet away. And what you can see is there didn't seem to be any correlation with, and this is the number of the mites coming into those hives, and there didn't seem to be a correlation between how far away they were from the collapsing hives and the mites coming in. Just the ones that are right in the middle of collapsing hives didn't seem to get any more mites than the ones that were further away. This is the total mite immigration over the trial from the middle of September till the beginning of October. And again, the highest we got was a little over 500 mites. Now, my guess is if we would have run this experiment the previous year, this would have probably been up in the thousands of mites, just based upon what we saw out in our alcohol washes in our breeder program. So unfortunately, we did it at a low mite count here. So on the average, we got 258 mites coming in each hive. But we also made the mistake. Let me see if I have it. No, I don't have it here. I'll show it next. So we also found that, and the donor hives didn't have magnetic traps, but there was plenty of drift between them because we'd open them up and we'd see bees of all color tags in those there. When the trial was finished after they collapsed, we took a magnet and we went on the bottom board and then the ground in front of those hives to see if any tags fell off. So this was a purple hive here, but we also recovered green and black and red tags, indicating the drift. So I eliminated all these tags from the count, and we recovered 3.5% of all the tags. Of the ones that actually drifted, that we got clear magnetic recovery, about a little over 2.5% of the tags we were able to recover. So we can go over those results now. So this is the percent of the tags recovered. So these are from the healthy controls. This one's a partial collapse. This is the full deformed wing virus collapse. And this shows you why it's important to run controls. We only ran 2, and I got lucky because one control had the least amount of drift of any hive in the experiment, and one control had the most amount of drift of any hive in the experiment, and all the collapsing hives were between those. If I had only run one control, my interpretation of the results would have been entirely erroneous, okay? So there was no indication that collapsing caused any increase in drift above the controls. So that was a very surprising finding right there. So then we looked at our recovery. So here's the home yard. At 15 feet, we recovered 7 tags. At 60 feet, we recovered 72 tags. At 500 feet, we recovered 66 tags. At a half mile, we got 11 tags. And this was actually the yard where I showed that alcohol wash there. So there was plenty of drift going there. We had traps on 3 hives out of 30. We recovered 11 tags, so if we would have had trapped them all, we probably would have gotten 110 tags recovered from there. And then we found 2 tags at this yard a full mile away. So the bees do drift for quite a way. This was our first tag we recovered, a white tag right here. And this arrow points to over this valley, over this hilltop to where my yard is there. This is not on a football field. This is very vigorous terrain around here. So it's not like a bee just gets confused easily and drifts a few feet. And you can see, so from the home here, they flew over a forest, over this valley, over irrigated pasture, and found this little tiny apiary hidden in the trees. And we got substantial drift from home to here at a half mile away. 6% of all the recovered tags we got from at this half mile away yard. So, okay, so here's magnetic recapture by date. And you can see that we had spikes of recapture. Again, differences in the distance right here. So I still have to analyze these collapse events and see if I can correlate the colors to those collapse to those collapse events. Here's the number of tags drifted to each of the 12 receivers. So this is the 15-foot, 60-foot, 500-foot range from 425 to 550 feet, half mile and a mile away. And what you see here is we got most of our recovery, not close and not really far, but in those mid-range right there. One would think you'd get most of your recovery very, very close. And then this is comparing the percent of tags drifted by the mite infestation rate at the time that we put the tags in there to see if there's a correlation. If the colonies that had more mites tended to drift more, and as you can see, there was no correlation, again, with our 2 controls being the 2 extremes. Okay, so now, is there a correlation between the recovery of the tag bees and the mite immigration into those hives? Here's the number of tags recovered in the colored lines, and then the blue is the total mite immigration for the season. So this hive here at 60 feet away was a bee magnet and a mite magnet. It had the most drift going to it of any hive and the most mites coming in. But this hive right next to it had very little drift of bees coming in, but a high mite immigration. So it was getting its mites from somewhere else other than those 9 hives that collapsed. And you can see that there was that we didn't have a strong correlation, that most of the drift came from hives other than those 9 collapsing hives. And there's no other commercial beekeepers, for 2 miles around. There's maybe a few hobby hives, but everything else, and all my hives for 2 miles around were all treated and close to varroa free. So where's all this drift coming from? I'm guessing it has to be from feral hives, escaped swarms, something like that is the best I'm trying to figure out. And when I did a correlation between the number of immigrated mites and the number of tag bees coming in, this is what's interesting. There wasn't a very good correlation, but the linear regression equation was interesting. What this says is this slope here indicates that for every tag bee coming in, you picked up about 7 mites. But there was a background level, 162 mites coming in independent of the tag bees. So there was a lot more drift coming in from outside mite drift than there was from my 9 collapsing hives. I found that really, really interesting. And when I do the math here, it suggests that nearly 1,000 bees drifted from the donor hives to the most attractive receiver. Okay, so that's a lot of drift right there. That was at 60 feet. The receiver probably got about 120 mites from the donors, but we recovered about 500 mites. So, so the 9 collapsed donors accounted for only a fraction of the measured mite drift. So it's coming from somewhere else. So this is what happened in 2017. Between the mid-September mite count in my breeders and the late October, we lost half the hive. Just this incredible. We went down from all these mite counts, of less than 10 in alcohol wash up to the, over 50, incredible immigration. We didn't see that during the year of this trial. The immigration was much lower, down around here. So I chose the wrong year to run this trial because I think we would have gotten thousands of mites coming into these hives. I have not analyzed the hive weight data, but here's one donor hive that clearly shows a collapse and perhaps robbing. And then here's a receiver hive that then shows a gain right here, but it didn't correlate on it. So I got to look at the rest of them. So here's the really interesting thing, is if you look at our 2 control hives, where their tags wound up. At 15 feet, 60 feet, 500, 2,500, 5,700 feet. One control, a whole bunch were very near, and the other one, not very many near. And then for all the 5 high mite hives, we got some drifting near, but most of the drift was further away, not in the hives that were right next door. So when I calculate the area covered here, you can calculate how many square feet there are inside this big circle or this one. And if it were random drift, say you're just dropping tags from above the donor hives, you'd expect most of the drift to be to the closest hives and very little drift to be out here. It's very easy to calculate the probability of just a random drift of a bee out here compared to a bee right here. So what I did is I calculated that probability here of the drifting. These hit 0 right after 60 feet, and for the chance of a bee just randomly drifting to that mile distance is you have a better chance of winning the lottery than a bee drifting a mile away. Here's the actual number of recovered hives. So compared to just the chance by area of drifting, and starting with arbitrarily just setting it at one-time chance for the receiver hive in the middle of the collapsing hives, 16 times higher chance of drifting 60 feet, 1,000 times higher of drifting 500 feet, 27,000 times higher chance, observed chance, going to half mile, and 144,000 times higher at a mile away. What this is indicating is that these bees aren't just getting confused next to their hive, they're drifting much further away. So your question, well, we'll get some answers here. So how much mite immigration into the hives is in the fall? There can be lots, at least 500 mites with no known high mite hives around, except for those few high mite ones there. And again, my guess is this would probably be in the thousands if I would have done this the year before. Do all the hives suffer equally from mite immigration? No. There's some that are mite magnets and some that, I don't know, they don't pick up mites from outside. Is it steady, the drift steady or episodic? There's a very large daily variation. So probably ask certain hives collapse or certain hives get robbed. Do the robbing bees carry back the mites? I have not yet analyzed that data yet. Do the bees drift equally from all the hives? No, not at all. Some hives, a lot of bees drifted, some hives they didn't, and it wasn't correlated with mite infestation rate. I assume, I'm testing the hypothesis, does deforming virus collapse increase the rate of drift? Well, this data does not support that, and I would have bet money and lost on this one. But we have that one control hive, mite-free, that had the highest rate of drift of anybody. Are some hives more attracted to drifting bees? It certainly seems so. There's some hives that just look like mite magnets, or bee magnets. And do bees tend to drift to nearby or faraway hives? This data very strongly suggests that they tend to drift more further away than closer. So I guess the take-home is, first we need to replicate this. So I've shown this technique. I'm hoping other researchers we'll use this technique and get better, more of this data so we can learn more about this. This is a lot of these answers we're getting here were surprising to me. So I just sent this article off yesterday to American Bee Journal. I'm calling for volunteers for a citizen science project for beekeepers across the country, and I'd love for some of you guys to do this, to just set up some hives, not with the tag, but just with the sticky boards and the mite strips inside, and go out twice a week and pull the sticky boards out, and collect data from all around North America and say, how much mite drift do we get into our hives during the late season? Because everybody's complaining about it, but we're just guessing about it. So I'm curious to see whether you have more drift if you're near commercial apiaries or near hobby apiaries. Down in the Bay Area, where we have all these treatment-free beekeepers, my guess is it's going to be an insane amount of drift, but I'd like to get quantification. So everybody's welcome. I'll post this to my website when it gets published to ABJ, and I'll supply the strips and everything, and I hope to get a citizen science project going on this. How am I doing for time?
You've got 5 minutes.
5 minutes. So we're going to do it really quick on the oxalic acid. There's my grandson. Every Californian grows up sucking on what we call sour grass, which is buttercup oxalis. So what you suck on is the very flavorful sap, which is a solution of oxalic acid and sugar inside the plant. And most of us grow up without too much brain damage from that, so it appears to be relatively safe, okay? Okay, so I did test Argentine strips with an absorbent cardboard, and my son said, Dad, in our operation that would take 12,000 strips. That's just too much time for us. So I did try the beekeeper standby, in the US at least, is the blue shop towel. Every commercial beekeeper has experience with blue shop towels in the US, so this was an easy first choice right there. I ran a couple of trials on this the first year, last year, and we got very good efficacy with the shop towels. But I had a lot of questions to answer. I'm going to go really fast. We made up a bunch of test ones, went out to a yard, laid them all out, 9 different formulations, different ratios of oxalic acid to glycerin, and 2 of them we tested propylene glycol instead of glycerin. Propylene glycol is a similar solvent, a food grade also, but it has a lower surface tension and lower viscosity, so I thought it might transfer better to the bees. We applied them, 240 25 hives in 14 different yards. And again, as Les was saying, you got to start with hives that have fairly high mites, but not so high that they're going to collapse and spread mites throughout your yard. So we started with all hives that had mite counts above 5 in alcohol wash and below 20 until we had a little bit higher. So fairly high alcohol wash counts. We found out that some of them, the ratios were easy to apply. One actually has a chemical reaction that happens when you put it out in the sun, and the towel itself gets hot to the touch and tears apart the cellulose, degrades it. So we're not going to use that with the propylene glycol. We also found that the high glycerin ones, using the same ratio as the Argentine formula, caused very much agitation of the bees, made a very quick mite drop, but a lot of agitation, and I didn't show it here. These bees in these hives bearded up in the front of the hives on the initial days. So too much higher ratio of glycerin to oxalic transfers the oxalic to the bee's body so fast that we get some brood mortality and causes agitation of the bees. I tested it at 21 days for a percent change in mite alcohol wash. If it's below the 1, that means that it went down. If it's above 1, it went up. And we found the ones that had half as much glycerin as oxalic, we didn't get much change early, and with propylene glycol, not. But the equal glycerin or double glycerin, this being the oxalic formula, or the Argentine formula, we got some initial mite drop. And this one right here is the very high glycerin one that just fast mite drop, but at the cost of irritation to the bees. We do neutralize the oxalic acid, just mix up a little bit of baking soda and water. The thing is, when you're using this, you can put it on your hands. You're not going to feel it at all. Maybe after 5 minutes or so, you'll feel a bit of warmth. So it's very safe to handle. But if you put on nitrile gloves, that reminds you not to pick your nose or rub your eye or something. So you have to train people. So like Thomas, one of our employees, he wanted to put the towels on at 1 yard, so we did. And when he was done, he walks back to the truck. But I kept my eye on him. and he still had the gloves on because the glycerin makes the oxalic acid really sticky. It touches everything. And as he reached for the truck handle, I yelled at him and I said, "Thomas, before you touch anything with those gloves on, would you mind going over there and taking a leak right now? Let me know how it feels." So you got, again, you have to watch your employees to make sure that they do safety. So we wash off our hive tool and our smoker. Very, very easy to neutralize. Very, very safe to use. We did also pull honey samples, send them off for testing. We waited 50 days for alcohol wash, and what we found was the control group went up 5.3 times in mite level. I used the Henderson-Stilton efficacy formula, which then tells you what your actual efficacy was compared to the controls. Every one of them was at least 80% efficacy under California conditions, and that's the same as we've seen the last 3 trials. So this one replicated that. Up to 99% efficacy on this particular formulation, the one-to-one formulation. The Argentine formulation over here was around the 90% efficacy. So we actually got better efficacy with the less glycerin on the extended treatment. We ran a similar trial in Georgia of just this one here. They got about 70% efficacy, but their control colonies were kind of weird. So I'm not sure why, whether it's the high humidity or what. So I can tell you in California, it works really, really well. I need more information from other areas to see how well it worked. But we probably have enough to run through getting it through EPA. Now in New Zealand, this is totally legal to apply. So what I did is on my website for the New Zealand beekeepers only the current formula and how to mix it up. So I do not suggest that anybody apply any illegal miticides, unregistered miticides to any hive whatsoever. I'm the only person in the United States who has a license to do this. I have my state offers me an experimental use permit every year to research on this. But for those in the rest of the world where it's legal, I am publishing what the suggested formula is. I did check also with the vaporization, since we were doing some experiments early on, and I'm doing some titration of bees to see how much gets on their bodies. I have a lot of bees in the freezer. I have to do this still, but each of these was a vaporization event from end of May to the 1st of September. So 9 vaporizations with the ProVape 110, and this is the alcohol wash count of bees in there. And you can see, if you think that you're going to get your mite count down with a couple of vaporizations during the summer when there's brood, you're dreaming. It takes a lot of vaporizations to get those mite counts down substantially. And so essentially, to get down to the level we got with one application of the shop towels, it took 9 vaporizations to do that. And in the shop towels, we had 18 grams of oxalic acid, much less, about one-fifth of what we used for the Argentine formula for a double deep. And these 9 vaporizations of 2 grams at a time. So this is also 18 grams total applied over time via vaporization. So the end result was about the same. One time you treated the hives 9 times, and the other way you would treat them 1 time. So this is the pros and cons. This is easy on the bees, one-time application. It is a slow action. It takes at least 6-7 weeks for full efficacy. And you do have some residues to scrape out. The bees don't tend to remove it, the towels when laid this way. Unlike if we drape them over, they tend to chew off the top edge, like with your strips, and they do remove them. Here's the ideal. You put them on when you put the towels on, when you put your honey supers on. When you pull your honey off, you got no mites in the hive. I mean, pretty cool, huh? So this is not yet registered, but this looks really, really promising. And thanks to my collaborators, Dr. Jay Evans from USDA, who is actually the registrant, USDA is, and Jennifer Berry and Jeff Williams over in Georgia. That's it. Good timing, huh?
Yeah, well,
Does it sound like have fun? I was a little bit overambitious this last summer, and my wife told me not again. This was 7 days a week from dawn till dark every single day. Just more research than any sane human being would take on. Yeah.
Very interesting. The question I have is on your queen selection talk there, you assume that the queens and drones share 50-50 with the chromosome load as far as mite resistance.
No, no, the queens that I choose supply all the genetics for their drones. So the genetics of the drones only come from the queen.
Yes.
So what you have is you're controlling half the genetics for the next queen comes from, or for the next workers come from the drones, and half come from the queens. That's what the 50-50 is.
My question is has there been research that shows whether or not those mite-resistant genetics are X or Y chromosome?
There's no extra Y chromosome in bees.
Oh, sorry. Yeah, no, I just wondered if the queen or drones carried more of those mite-resistant.
No, every bee is by default a male. Only when you have two different sex alleles on the chromosomes that turns on, activates the gene called feminize, which then turns into a female. So if you don't have two different alleles at the sex chromosome, every bee turns into a male. That's the default. So there's no difference in an egg between a male bee, a female bee, a worker bee, a drone. All the eggs are the same. It just depends upon how many at the sex determination gene, whether you have one or two different alleles. Okay? That's a simple question.
On your mite.
Where are you? There you are.
Yeah. On your control hives and on your collapsing hives.
Yeah.
Was it the same genetic makeup of those hives?
They, yes, yeah, they actually were all carniolans because those were supposed to be the black ones. Okay, so they were, and then the receivers were Italian hives. Yeah, so exactly the same genetics, same queen stock from the same breeder. Yeah. What was I going to say? Oh no, this thing. Oh, on the breeding program. One of the things, the question is, wow, when you select for these mite-resistant, apparently mite-resistant, we call them apparently mite-resistant queens, are they good bees? And my sons were going through we moved most of them to a single yard before almonds, and they were grading them before almonds. See, I go back and forth when I'm talking to an almond or almond audience. We were grading them, and Ian goes, "Dad, get your camera over here. Look at these hives. Mite counts as 0, and these are the best hives. Some of these are the best hives in our operation. Big booming hives, strong, beautiful brood, lots of honey producers." So I'm not seeing any negative effect for selecting for varroa resistance. And that's really a positive thing. Yeah.
Last one.
Okay. With regard to your study that you did with the oxalic acid,
Yeah.
you mentioned you took samples of honey.
Yeah.
What did you actually test the honey for? Like, what were the
For
residues?
oxalic specifically,
Okay.
you have to talk to Jay Evans. He bought the kit from Europe somewhere, so he would be the one who could answer on the testing. I didn't do the testing.
And just to follow up on that would be, is there any concern about when you apply acid to a hive all the way through that honey flow, whether it might be contributing to a lot of HMF in that honey that now is going to be saleable? And some markets may be...
That's an excellent question, Rial. We did not test for that. Yeah, that'd be a good thing to test for. The thing is that oxalic acid is a natural component of honey. And what we found is it did not increase that appreciably. So I would tend to doubt that it would cause any increased HMF because honey is already acidic, and we're only touching it, down in, minute decimal points. Okay.
Yeah. But I think the drizzle of 3.5% won't have much of an effect. But I'm just wondering with that Argentinian one where the concentration of oxalic acid is fairly high that it might.
Yeah, and that's why I'm not crazy. For us to put 4 strips in a double deep did not give us the good results. It took 8 strips in a double deep. You're doing single brood chambers. In our double brood chambers, it took 8 strips to get good efficacy. And that's 80 grams of oxalic acid. 80 grams going in there. I'm using 18 on the shop towel and getting equal efficacy. So the delivery method is showing me a lot. Now that 80 grams doesn't all go into the hive because when those strips come out, they're still full of oxalic acid. If you don't believe me, stick one in your mouth. So you'd have to subtract, you'd have to then titrate the amount of oxalic acid in the strips that are removed and then subtract that, and that'd be your actual delivery. But the same thing with the shop towel. So I say I'm putting 18 in, but not all 18 are going out there. Although when you do put the shop towel in your mouth, there's less acid than there is in the Argentine strips.
All right.